about bioreduction, an alternative strategy for storing fallen stock prior to disposal, or to put it a bit more simply, turning dead animal carcasses into a liquid waste that can be disposed of at a time suitable for the farmer. So my PhD was funded by a British pig executive, but it also forms part of a large project which is also sponsored by the Welsh Government and Hubby Cymru, the meat promotion service in Wales. So in this presentation, I'm going to give you an introduction as to why we need a novel method of storing animal carcasses. Then I'll explain a bit about what bioreduction actually is and how it works. Then I'll go over the thesis aims and how we meet these aims in the individual chapters, before going on to summarising conclusions and some recommendations for future work. So in the UK, we have a livestock population of close to 210 million animals. With mortality rates of between 5 and 10%, this means that a large amount of waste is generated on farm and needs to be disposed. Traditionally, disposal would have been by burial or burning, but the introduction of the Animal Byproducts Regulations in 2002 restricted these methods of livestock disposal because of the risk of BSE transmission and the spread of other diseases such as foot and mouth disease. In response to this, the UK government set up a national central collection service. So when an animal dies on farm, you ring up the central collection service, who comes and picks up your dead animals as part of a milk round system. However, the livestock industry in the UK has raised concerns about this because disease has been linked with transport of animals in the past, such as foot and mouth disease, which is what you can see in this picture. So collection vehicles may pose a biosecurity risk. So the industry put out a call for novel methods of livestock storage or disposal, and this is where bioreduction comes in. Bioreduction is an on-farm method of fallen stock storage, which could be biosecure. So what actually is bioreduction? Well, bioreduction can be defined as the aerobic degradation of animal byproducts in a partially sealed vessel containing water with contents that are heated to 40 degrees and aerated. So we have these bioreduction vessels which have a capacity of 6,500 litres. They're made of a thermostable fiberglass and resin mix. So when your animal dies on farm, you make an incision into the abdomen of the carcass to release the internal microorganisms and enzymes, and then these carcasses go into the vessels which contain water. However, it should not be mistaken for anaerobic digestion. These vessels are actively aerated using air at a rate of 50 kPa for 45 minutes of every hour. In theory and in practice, bioreduction reduces the amount of waste to be produced because a lot of the water from the carcass disappears as evaporation. So the primary aim of this thesis was to provide evidence to the European Food Safety Authority on the biosecurity of bioreduction. So EFSA are the organisation responsible in the EU for saying whether novel methods of disposal or storage will or will not be allowed. Secondary aims include determining the efficacy of the ingested product, and I'll explain what the ingested product is further on in this presentation. We also wanted to see whether bioreduction is environmentally friendly, and also to try and understand the biochemical and microbial cycles so we can try and improve the efficiency of bioreduction in the future. So if we start by looking at biosecurity, EFSA has said that a novel disposal technology will be allowed if we can show we have a 5-log reduction in thermotolerant bacteria such as Enterococcus faecalis or Salmonella septemvir, or a 3-log reduction in thermotolerant viruses such as the model virus porcine parvovirus. However, we're proposing bioreduction as a novel storage technique, and the updated regulations in 2009 stated that for containment, pathogens must shown to not proliferate. So this is the study site we have out at the University Farm. At this site we ran 2 trials. The first, trial A, under standard operating conditions. So carcasses were added to the vessels as and when they died on farm. The vessels were heated to 40 degrees and they were aerated. And the second trial was run under simulated breakdown conditions, so the vessels were switched off. 3 of the vessels were used as treatment vessels, which we inoculated with pathogens. 2 were used as control vessels, so although carcasses were added, we didn't actually add any pathogens. And all 5 of these bioreduction vessels fed into the compost wood chip biofilter to try and reduce odours. Samples were taken from the liquor that you get from the decomposition of the carcasses, from the bioaerosols being released from the vessels, and from the biofilter. I'm going to talk to you in this presentation about the results from the liquor, but obviously if you want more results then I can give these to you at another time. Studies were run for 127 days, which is just over 4 months. So this is what bioreduction actually looks like. The photos in the top row are from the trial run under operating conditions. So carcasses were added as and when they died on farm, and the row on the bottom shows the carcasses added when the vessels were switched off. So the main difference between these is that as decomposition occurred, by day 28 we actually got a reduction in carcass material. This carcass that you can see is actually a fresh addition of carcasses. So the majority of carcass waste was decomposed in 28 days. And then we can see further additions of carcasses as space has become available in the vessel. However, in trial B, we have serious retardation of decomposition. So these 3 carcasses are exactly the same carcasses as those 3 at the beginning of the trial, and very little decomposition has taken place. So if we have a look at the bacteria that EFSA required us to look at, so we've got Salmonella on the top and Enterococcus on the bottom. The red bars represent the number of bacteria that we can count on agar plates at the start of the trial, and the cream columns represent the numbers of bacteria that we can count at the end of the trial. So from these results we can see that we achieved a 5-log reduction in both Salmonella and Enterococcus when the vessels were run under standard operating conditions. However, for trial B, when the vessels were switched off, we can see that bacteria still survive until the end of the trial. So we can say that pathogens are contained within the vessels because we have reduction in numbers. To look at viruses, our partners over in Northern Ireland used a technique known as quantitative PCR. So they looked at the number of genome copies of the virus, porcine parvovirus, in the liquor samples. And from this data, it does look like we have bacteria surviving until the end of the trial, both under standard operating conditions and simulated breakdown conditions. However, looking at DNA does not tell you whether the DNA was from viable or dead cells. So they also looked at the infectivity of pig kidney cells. And infectivity is the important thing. And indeed, when the vessels were switched on, we did see a 5-log reduction. So this is a 2-log reduction greater than that which EFSA required. However, when the vessels were switched off, we still see that the virus persists until the end of the trial, but again, there is no proliferation. So from EFSA's viewpoint, we can say that bioreduction is biosecure. Previous work done on bioreduction has used an ingester project known as Gel 60. The active ingredient in Gel 60 is sodium alginate, which is derived from the brown seaweed Ascophyllum nodosum. And it's thought that the matrix of polysaccharides, fibres and proteins acts as a catalyst for microbial growth and activity, which encourages microbial activity and encourages them to break down the more recalcitrant material of the carcass. So it's supposed to increase the rate of decomposition. However, this has not been scientifically ratified, so we wanted to have a look into this. So to do this, we looked at some other commercial accelerants that were available to us, and we used mainly bacterial inoculants because these are very popular. So we used Filter Boost, which is used in biofilters in fish tanks, and we used 3 different chemical accelerants from the Alpine Murray Clear range, which are used to break down grease in things like wastewater. We also looked at liquor from a previous bioreduction experiment. So for things like compost and anaerobic digestion, you usually use inoculum from previous digestion to stimulate growth in fresh batches. And we use soil to see whether that would work as well. So we set up these mini microcosms which contained pork and water and either low dose or a high dose of accelerant, with the hypothesis that the mass of the microcosms and the moisture content of accelerants that were working would be less than the control, and protein and amino acid contents of accelerants that were working would be higher than the control. Unfortunately, or fortunately for us, none of the experiments tested actually met all of the requirements. And indeed, when we used gel 60 at the high dose, which met some of the requirements, we used it at a rate of 500 times higher than that recommended by the manufacturer. And still, it didn't work sufficiently. So we could conclude that these experiments were not suitable. So going on to the carbon footprint, the primary aim of this work was to meet one of the Welsh Government's objectives. So in the policy Farming, Food and Countryside, they stated that greenhouse gas emissions from novel disposal methods would be low. Also, when we consider farming as a whole, greenhouse gas emissions are often or always missing from life cycle assessments. So we identified this gap in the information that we wanted to fill. And then we could use the information gleaned from the carbon footprint to identify areas for improved vessel design. So to do this, we looked at 5 different storage and disposal scenarios based on a study that was done at Henvice in the past. So we looked at the burial of carbon. We wanted to look at the greenhouse gas emissions from the death of the animal right through to final disposal. So we looked at burial. So although it's not allowed under the EU regulations, it is used in other parts of the world and in isolated parts in the EU as well. We also looked at conventional disposal, which is the central collection service. The DOLAV option is similar to the central collection service, except that you can store your carcasses in an airtight, watertight box, and therefore you can leave it slightly longer before your carcasses are collected. Freezing has been used in previous disease outbreaks, and then obviously we wanted to look at bioreduction. So for burial, the carcasses don't go very far. They go from the burial site, go from the fields where they were found to the burial site. The central collection service, the DOLAV method, and the freezing all go to a central collection point on a farm before they're picked up and taken away. For bioreduction, the liquid is taken out of the vessels via a suction pump on a waste truck, and this is transported off farm. Because of the animal byproducts regulations, this waste must either be rendered or incinerated. And just in case bioreduction was ever allowed as a disposal option in the future, we also looked at whether replacing chemical fertilisers would reduce the greenhouse gas emissions from bioreduction as a whole. So just looking at the results from this, unfortunately, bioreduction seemed to have the highest carbon footprint. But when you get down to specifics, it's not really surprising. The bioreduction vessels are run off electricity, and in fact electricity accounted for 99% of the carbon dioxide equivalents emitted per year. But it's not all bad news. We can mitigate against this excess electricity usage. For example, we've shown that when vessels are switched off, pathogens are still contained within the vessels, and therefore they're still biosecure. So we may be able to switch vessels off during periods of activity, such as the lambing periods. Also, the bioreduction vessels are run at 40 degrees because this is similar to the temperature found in live carcasses, so it's optimal for the microorganisms and enzymes. However, if we reduce the temperature by 10 degrees, this may not have a big effect on the activity of the microorganisms or enzymes, but it will have, or is hypothesized to have, a big reduction in electricity savings. At the moment, the vessels are single-skinned, so it's very important that future vessels are double-skinned with a layer of insulation in between, and this will keep the water and the carcasses heated for longer, so you need to use less electricity to heat the vessels. And also we could look at renewable energy. However, despite having such a high carbon footprint, put in context, bioreduction still has a carbon footprint of less than 2% of the whole farm carbon footprint, so it's still a relatively small amount. So the final chapter we looked at was understanding bioreduction, so what is actually happening within the vessels. And to do this, we wanted to look at the physicochemical cycles within the vessels. We wanted to identify major enzyme families that were involved, and to look at the microbial communities. So in this trial, we used 2 vessels, and we added 150 kilograms of pig carcasses to the 2 vessels. That is actually a pig, not a sheep. And we ran this experiment for 56 days. So we studied 8 enzymes to look at activity: 2 acetylesterases, a lipase, an aryl sulfatase, a phosphatase, glucosidase, protease, and dehydrogenase. The 2 enzymes that had their highest activity were the 2 acetylesterases. And I just want to emphasise this by showing you the differences in scale on the y-axis. But I also want to emphasise the fact that we have differences between the 2 vessels. So the closed circles are bioreduction vessel 1 and the open circles are bioreduction vessel 2. So although we're doing the same thing to the carcasses, we've got similar amounts of carcasses, the enzyme activity is actually different between the vessels. So now if we go on to physicochemical parameters, the average pH of the pig trial was 8.65, so the liquor was moderately basic. And this isn't a surprise, this is what it was like for sheep as well. But we did have 2 distinct pH phases, so we've got the 2 bioreduction vessels again, and after approximately 28 days we went from a reduction in pH to an increase in pH. And it's hypothesized that as the aryl sulfatases broke down the fats, it released released fatty acids, which reduced the pH of the liquor. And then as the lipase took over from the aryl sulfatases, these broke down the fatty acids even more, which increased the pH again. For nitrogen cycling, I know a lot of people do nitrogen cycling, we assumed ammonification as the primary end route. We had a lot of ammonia in these vessels. For nutrients, particularly, so ammonium nitrate and phosphate, concentrations were significantly higher in bioreduction vessel 1 than they were in bioreduction vessel 2. So we could say that there are differences in the physicochemical properties between the vessels, similar in the way that we have differences in the enzymes. But we also have a difference between species. These average quantities you can see on your left are considerably higher than the nutrient concentrations we had for sheep. This may be because this trial was run for slightly shorter amount of time, so if we run it for longer, then the means may have reached a comparable amount, but we'd have to redo that experiment in the future. So to look at microbial communities, we use a technique called a riser, and when we do a riser, we get a really complicated picture that looks like this, which is a non-metric multi-dimensional scaling analysis. Try saying that when you hear this. So just to note, the triangles are bioreduction vessel 1 and the circles are bioreduction vessel 2. And I just want to point out at the start of the experiment, which is the blue ones, we can see that the 2 microbial communities are quite far apart. So this suggests that at the start of the trial we have different starting inoculums. However, as we progress towards the middle of the trial to days 28 and 42, these communities converge. So we have a climax community. However, for some reason or other, the communities then diverge until the end of the trial, which are these dark red ones, so they are quite far apart. So again, this just emphasises the difference between the vessels. So to go on to conclude, to make some general conclusions, we can say that the bioreduction of sheep is biosecure according to EFSA's requirements. However, we did get some foaming in the pig vessels, and therefore we need to make sure that that pig liquor is biosecure. The experiments tested did not speed up decomposition, but this doesn't mean that we can't produce our own starter inoculum in the future, designed specifically for bioreduction. The electricity usage greatly impacted on the carbon footprint of bioreduction, but it still represented less than 2% of the whole farm carbon footprint. Bacterial communities within the vessels are different and depend on the stage of decomposition of the carcass inputted, what has previously been bioreduced, and the age, weight and size of the carcass and the tissue composition of the carcasses. For example, if you have a carcass with a higher fat content, that will decompose quicker than a carcass with a lower fat content. I really want to emphasise that simple changes to the bioreduction vessels could vastly improve the technology. So this leads on to recommendations for future work. It would be really beneficial for bioreduction if it could get accepted as a disposal technology. And to do this, we would need to look at the biosecurity associated with land spreading, so it would be a completely self-contained disposal option, and also the biosecurity of pig carcasses. From a practical point of view, there are several things we can do to improve the vessel design, such as reducing electricity, so we may want to look at reduced water usage, reduced temperature, and reduced aeration rate. Because we had a few problems with foam for pigs, we may want to look into some anti-foam technology. And then what is most exciting for the future, in my opinion, is to generate a starter inoculum. And to do this, we would need to identify key enzymes and the key bacterial and fungal genera involved in bioreduction. So finally, I just want to say thank you to Professor Davey and Gareth for giving me this opportunity. Also to Ceri, because she helped me do a lot of the GUI work in the biosecurity trials. And to everybody else who helped out along the way, and especially to BPEX for funding this work. Any questions?